Hey guys, so today we're going to actually be getting into the evolution portion of this chapter. We're going to be looking at all the different evidence that we have that evolution is actually happening. Okay, so for evidence of change, we're going to be looking at stuff like biogeography, radiometric dating, and then things like morphology, embryology. Essentially, you need to know all the different ways that we determine that species are evolving and how old those species are. So how far, how long ago they evolved. So biogeography is the study of the variation and distribution of life on Earth. So where are different organisms found on Earth? That's the distribution. And the variation is how different all of those different organisms are. By analyzing both the past and current patterns in this distribution of species, we can kind of start to see how those species evolved over time. This is because Earth's land masses have undergone such major changes because of continental drift. So back 225 million years ago, all of the continents were part of one giant land mass, which you guys probably already knew. And then slowly the tectonic plates pulled those different land masses apart until you get the positions that they're currently in. So that's important because species that evolved before 150 million years ago, we can kind of see that their distribution is across more than one of the land masses that we have here today. So if a species appears in the fossil record of multiple of our current land masses, then that species probably evolved before those land masses separated. If the species are only found on one of our current land masses or in the fossil record of one of our current land masses, then it probably evolved after that land mass split off from the other continents. Species that are endemic to a certain island likely evolved on that island from some sort of founding ancestor. Endemic meaning that the species is only found in that one location and nowhere else on Earth. So this is kind of an example of that. So this fern here is found in the fossil record of Australia and Antarctica and India and Madagascar and Africa and South America. So it almost certainly evolved when we still had that supercontinent. That's the only way that that fern could be found in the fossil record of all of these different continents, land masses, whatever you want to call them. And so as soon as we see this sharing of a fossil record across multiple land masses, we know that that species evolved before the separation of the land masses. Okay, radiometric dating is a even more accurate way, a much more accurate way of determining the age of a species in the fossil record. Essentially, we're using radioactive decay to date the age of the rock that species is found in. Isotopes decay naturally at a constant rate, so we can determine the age of the rock or the age of the fossil based on how much the isotopes have decayed in that sample. Radiometric decay has been used to date the Earth as a whole, as well as individual fossils that we found. The oldest fossil discovered to date was dated at about 3.8 billion years old. Okay, so that's kind of how we date our fossils, figure out how old they are. But we're going to be looking at not only fossils, but a bunch of other ways that we essentially know that evolution is happening. So all of this is evidence of evolution. Evolution is when an entire population or species changes to become something new or distinct over time. We define it as a change in the gene pool of the species, which is the genetics of the species changing. And this is all of the different types of evidence that we're going to be looking at. So fossils, genetics, biochemistry, morphology, embryology, vestigial traits, and just observation. So paleontology is the study of fossils. We have discovered about 250,000 different fossilized species. Fossils can include remains, which are like skeletons, that's what we often think of when we're thinking of fossils, but it can also include impressions, which are things like footprints, as well as traces, which are things like egg and feces, anything that would be left behind by an organism, but not the organism's body or skeleton itself. Most organisms, unfortunately, never fossilize, and this could be due to their body structure. So if an organism doesn't have a skeleton, it's much more difficult for it to fossilize. It could also be because of their habitat. If the habitat is somewhere where there's like a lot of, or there was a lot of like mud or volcanic activity or something to bury the skeleton after the organism died, that made it much more likely to fossilize. If it was just going to be like left open to the air and the elements, it probably wouldn't have fossilized. And so because of this, only a very small percentage of the species that existed on Earth will ever be found in the fossil record. But fossils do prove that we have had a lot of different species that have lived here on Earth at different times. Most of them are now extinct. And it also shows the complexity of living things have increased over time. So the further back we go into the fossil record, the more simplistic those organisms are. Living species are usually located in the same region as their most closely related fossilized ancestor, kind of supporting this idea that the living species evolved from that fossilized ancestor. Usually, if you find a fossil, you're not going to find a living species that is similar to it on like a completely different continent. Okay, the second type of evidence is genetic evidence, which is gathered by analyzing the sequences of DNA. DNA is the hereditary material that contains all of the genes which code for all of the different traits of that individual. DNA is made up of nucleotides, which contain specific nitrogenous bases. So essentially, DNA is made of ordering this A, T, G, and C in different orders. So it might be like A, T, C, C, G, or something like that. And the order of these bases can be analyzed to identify the relatedness of species. The more similar the order of those nucleotides is, the more closely related the species are. This is pretty much the most accurate way of determining species evolutionary history, because we can kind of differentiate between analogous and homologous traits. Essentially, it means that we're not going to be tricked by organisms that look alike but aren't related. Okay, then we have biochemistry, which are proteins that are going to be analyzed. So proteins are molecules that make up lots of the different pieces of an organism, everything from fat to muscles to skin, your eye, everything is made up of proteins. Proteins are made up of amino acids that are joined together in a specific order. And so if we just take the proteins of different species and look at the order of their amino acids, the more similar those different amino acids are, the more related the species are. So that's what this example up here is showing. It's essentially talking about the number of differences in that amino acid sequences between different species. So we have like a human and a monkey might only have like 8 differences. A human and a dog might have 32. A human and a bird would have 45. There's 125 different between a human and a sea lamprey, which is this like eel type thing with like a circular mouth. Anyway. So yeah, you're essentially with genetics, you're sequencing the nitrogenous bases, we're sequencing the nucleotides of DNA. With biochemistry, you're sequencing the amino acids of a protein. But they work the same way. Then we have morphology. This is the method that used to be used primarily but has fallen out of favor. Morphology refers to the physical structures and skeletons of a species, and similar morphology can indicate relatedness, but it can also be deceiving. So homologous features refer to structures that are similar in appearance, might not necessarily function the same way, but they do come from a common ancestor. That is the important part about homologous features, is that they are on species that are related. However, you can also have something called analogous features, which look very similar and probably function the same way, but are not at all evolutionarily related. They can trick you into thinking the species are evolutionarily related because they look the same, but they're not. So these are some examples. So at the top here, this top example is some analogous traits. So for example, the fin of a fish and the fin of this penguin and the fin of this dolphin, they all look the same from the outside at least, and they're all doing the same thing. They're all propelling these organisms through the water. However, one is a fish, one is a bird, one is a mammal. They are not evolutionarily related. So the fins of these different species are analogous. They function the same way. From the outside, they look the same. They are not related. And then on the bottom here, we have our homologous traits. These are ones that from the outside look really different, though the skeletons are actually kind of similar. Very different functions, but they are actually related. So we have the arm of a human, we have the leg of a cat, the fin of a whale, the wing of a bat. The different color coding parts here are showing you that we have the same bones. They're just kind of elongated or shrunk. But obviously these all function very differently. One's an arm, one's a leg, one's a fin, one's a wing. But all of these, humans, cats, whales, and bats, are all mammals. So they are all related. So homologous traits are related, might not look related. Analogous traits are not related, even though they might seem like they are. So because of analogous and homologous traits, this is why we don't really trust morphology anymore. But back before we had the technology for biochemistry and genetics, it was the primary way of kind of guessing how species were related. Okay, embryology refers to the study of embryonic developments of different species. Again, it's just kind of a way to determine the evolutionary history of a species, so what kind of the ancestors are. Species that have similar embryonic structures tend to be more closely related. And if during embryonic development, you have structures that develop and then disappear, or develop and are then modified, it can show a structure that our ancestor used to have. So for example, human embryos develop tails, which shows us that we had an ancestor that had a tail. Human embryos also develop gill slits, which shows that, you know, back in the deep recesses of our evolutionary history, we had an ancestor that had gills. The tail obviously disappears and we're left with just the tailbone. The gill slits are actually modified into what is now our inner ear bones. And the gill slit modification also happens in chickens, which tells us that humans and chickens have a common ancestor that both had gill slits. Now, that's not to say that we're closely related to fish or closely related to chickens. We're not. It just means if you go far enough back into our evolutionary history, really pretty much everything evolved from something that had gills at one point in time. Things evolved in the water before they ever evolved on Earth. And the things that evolved in the water eventually developed limbs, and then eventually developed lungs and could move on to land. But all of us, if we go back far enough, all species, if we go back far enough into our evolutionary history, came from aquatic organisms. So this is just kind of what this looks like. So yeah, you can see that all of these different species, fish, salamander, tortoise, chick, rabbit, and man all develop these gill slits. And that's because, again, we all have that common aquatic ancestor. But then as we develop further here, you can start seeing, okay, if we're looking at the human, it looks a lot more like the rabbit and the chick and the tortoise than it does the fish and the salamander. And then if we go on even further into the embryonic development, that human looks a lot more like the rabbit than it does the chick or the tortoise. And all of that makes sense. We are more closely related to our land-dwelling, other land-dwelling species than to aquatic species, and we are most closely related to other mammals compared to like birds or reptiles. Though I do think the tortoise has the cutest embryo. Okay, vestigial features are physical features that have no function. They also tell us something about our ancestor. Vestigial features always had a function in our ancestor, and we have just evolved to no longer use that feature. The current species doesn't use it. We have lost the function of that feature, but not the structure itself. So examples of this are whales and snakes, which have vestigial hip and leg bones, but obviously snakes don't walk, neither do whales. They don't use those pelvis and leg bones anymore, but they are still present because both whales and snakes evolved from things that walked with four limbs. This is an example of that in a whale. It's just kind of hidden under the blubber. You would never know it was there unless you found the skeleton of a whale. With humans, our goosebumps are an example of a vestigial feature. When we get cold, we tend to get goosebumps. Our ancestors would have used that to insulate us back when our ancestors were like fully covered in hair or fur. The goosebumps would puff up the hair to provide insulation and warmth. We no longer have enough hair to make that useful, but it still happens. So it's a vestigial feature. Okay, and then lastly, we have observations. Evolution usually occurs so slowly and gradually that it's difficult or impossible to actually observe happening. But there are some exceptions to that. If there is a really strong selective force, which is something changed and it's killing off a large chunk of the population, so that could be a major shift in climate, a natural disaster, a disease, something that's really going to limit the food supply. When you have something like that happening, the evolutionary changes can happen much more quickly. So scientists have been studying a group of finches over, I think, like 6 or 7 decades now. And anytime the environment there shifted really dramatically and it affected their food supply, these scientists actually observed the birds evolving, usually changes in their beak. So their beak size or shape would change based on the availability of food, which was based on the climate shifting. We can also see evolution under artificial circumstances. So we can essentially use artificial selection to cause an organism to evolve a trait that we like. So we've done this with tons of different plants. We've done it with dogs, cats, a lot of like farm animals, stuff like that. Essentially, you take, if it's like crops, you take like 2 plants that both have a trait that you like, and you breed them together, and then you take their offspring that have the best traits and you breed those ones together. And you just only breed the ones that have the traits you desire, and eventually you have evolved artificially that species into something new. So this is an example of a type of evolution that humans have done. So we have taken this sea cabbage plant, and through artificial selection, we have evolved it into all of these different types of plants, which now all have this common ancestor of the sea cabbage. Okay, so that is it. Make sure you're familiar with all these different types of evidence of evolution, and let me know if you have any questions.